delighted to have Philip Ball all the way here from London. He's really one of the world's great science writers. He's really written on so many aspects of science, from chemistry, biology. He's famous for his book on patterns. When it first came out, it was called Nature's Tapestry. And now it's since been spun off and expanded and revised into 3 different books: Shapes, Branches, and what's the third one? Flow. All right, flow. Flow, shapes, branches. Probably right here. It's not. So anyway, there's an interesting dichotomy going from the morning session to now, in that in sexual selection in biology, it's often emphasized anything could be preferred by females of the species in all these cases. They could choose to like anything, any sort of weird thing. Is that really true? Or there's certain patterns that nature has at a deeper root within chemistry, within physics, within mathematics itself. Are these the patterns from which beauty and the aesthetic are chosen from? So I think in the next session, starting with Philip and then with Tyler, we're going to delve down deeper into the patterns of nature at a fundamental level. So I'd like to welcome Philip Ball. Thanks a lot.
Thank you very much. Is this, let me bring that up a bit. This audio feedback, is that okay? I knew this would be interesting. That's why I thought it was worth coming across the Atlantic, and it certainly was. Thank you very much, David, for making that possible. Okay, I'm going to start with the basics. Let's go right back to this question. Why is a zebra stripy? Well, the answer is obvious, isn't it? The stripes help the zebra hide amongst foliage. The British zoologist Hugh Cott, who was a specialist on camouflage, quoted one observer as saying, "In thin cover, the zebra is the most invisible of animals." And besides this, we have that most dependable of sources, Rudyard Kipling, to back up this idea. In his Just So stories, Kipling gave this as the genesis of the zebra's stripes. He said, "After another long time, what with standing half in the shade and half out of it, and what with the slippery, slidey shadows of the trees falling on them, the giraffe grew blotchy, and the zebra grew stripy, and the eland and the kudu grew darker, with little wavy grey lines on their backs like bark on a tree trunk. And so, though you could hear them and smell them, you could very seldom see them, and then only when you knew precisely where to look." So you can see that even Kipling was a budding Darwinist in his implication that the zebra's stripes must serve a useful purpose, even if his explanation is here rather more Lamarckian than Darwinian. There's only one problem with this explanation, and that is that the zebra's normal habitat doesn't actually look like this. It looks more like this. So it's not at all clear what these stripes are about. And in fact, it's not that we're puzzled by why this is so. In fact, we have too many explanations. So why is the zebra striped? Is it camouflage, or is it some of these other explanations that have been put forward for why the zebra has stripes? And this last of them, actually, protection from biting flies, such as the tsetse fly. This is a very recent idea, or at least it's been very recently supported by some evidence in this fantastic experiment. This was an experiment done not by biologists, but by physicists. I think this is a physicist's idea of a biological experiment. So they have these model horses that they've painted and then covered with fly glue and stuck out in the fields in Hungary, as it happens. But there are biting flies in Hungary. And they found that the flies seem to be less attracted to the stripy model than to the others. They just count out how many flies have stuck to them, and they find this. So actually a very nice experiment, and it seems to have something to do. It seems that flies are repelled by stripy surfaces, and that has something to do with the way that reflected sunlight from these different surfaces gets polarized. Well, the point is that if you assume that the zebra's stripes are a Darwinian adaptation, that they somehow contribute to the zebra's fitness, then there's apparently no end of the just-so stories that you can spin about what benefits they might confer. Now, I should make clear that I do suspect that the zebra stripes are adaptive. I suspect that there is some sound evolutionary reason why they're there, even if we don't really know what it is yet. However, there are three warnings perhaps, that I want to add to this story. The first is the objection that was raised by to this kind of explanation, this kind of adaptive explanation, by the Scottish zoologist Darcy Thompson in his classic book of 1917 on growth and form. This is Darcy Thompson here. We saw pictures of Wallace and Darwin a little while ago. They're all kind of interchangeable. It makes you feel like beards going on at the end of the 19th century. What Darcy Thompson said, or at least one way of reading this fantastic exploration of his of shape and pattern and form in nature, is that there are constraints imposed by the laws of physics and chemistry, which not only limit what biology might achieve, but also often provide a more immediate and satisfying explanation for what we find in the living world than invoking Darwinian origins. Now, it's not that the Darwinian explanation is wrong, or not necessarily that, but that it's incomplete. If we're content to say that we've explained the zebra stripes, if we can show that they somehow have some fitness benefit, we still have failed to account for how these markings get put in place on any particular zebra, because it seems clear that the precise location of each dark band isn't fixed, isn't determined by genes. In other words, evolution might provide the why, but not the how of these stripes. Secondly, to assume that everything we see in the living world is an evolutionary adaptation it's a reasonable starting point, but it can become a lazy reflex that simply forecloses any further inquiry. Have a look, for example, at these patterns on mollusk shells. You see they're wonderful, they're hugely varied, and yet if we're going to look for adaptive explanations for them, then we start running up against some uncomfortable truths. Many marine mollusks spend essentially all of their lives buried in mud. So you don't see these patterns. Others have these patterns on their shells, but then they cover them up with an opaque coat of some kind of organic tissue, almost as though they're embarrassed by their own virtuosity. Other kinds of mollusks have shell markings that individually might vary from, between one individual and another within the same species, with such variety. Here's an example from a single species, that you can't really imagine how any of these species would recognize the other one as coming from the same species. Now, it's not impossible that even here that there may be an adaptive explanation for these pigment markings in the shell. For example, it's been suggested that these bands and these spots and stripes might incorporate unwanted waste products and so lock them away from the surrounding environment, from the water surrounding the organism itself, these toxic waste products. But even then, if that's so, the patterns that result are largely a chance byproduct of that process and are subject to very little, if any, adaptive evolutionary pressure, selective pressure. And under those circumstances, it's not fanciful to say that nature is at liberty to play, that it has something like an irrepressible impulse to create patterns, and that it does so even when there's no evolutionary need for them. And this leads me to my third point. Even if we accept that the zebra stripes have an adaptive function, we can't help but be struck by how they resemble some patterns that evidently don't have an evolutionary function to them, because they form in non-living systems. And this seems to be a common theme in the patterns that we find in nature, that they come from a limited palette. We see the same patterns appearing again and again. So here are some other examples of stripes in nature. We see a lot of hexagons and honeycomb shapes in living and non-living systems alike. Spirals are very common, and branches. Are these resemblances just coincidence, or is it something more? Well, I want to suggest to you that, at least in the case of the zebra stripes, and in several others in the living world, the resemblance between these patterns and others we find elsewhere in the natural world probably aren't coincidental at all. Now, resemblances like this were noticed by Darcy Thompson. In fact, that was one of the key themes of his book. One of the patterns that he considered very briefly in his book, and this is taken straight from his book on the left-hand side here, are these patterns formed in a chemical system. And these structures are caused by the periodic precipitation of a coloured salt formed by the reaction of two compounds that have been infused into a gel in these columns. And as the reaction proceeds, you get a sequential appearance of these bands. And on the right-hand side, I just show a modern example of this experiment where they're slightly clearer. These are known as Liesegang rings, or Liesegang bands, after the German chemist Raphael Eduard Liesegang, who discovered them in 1896. Darcy Thompson suggested that perhaps they might be related to the banded mineral formations that we see in agate and onyx. And some others suggested that these bands and stripes might represent a simplified version of the markings that we find on zebras and on tigers and on some butterflies. Now, that seemed to a lot of people a rather fanciful leap at the time, but it turned out to have more than a grain of truth. And the reason why emerged from work in a quite different area of chemistry several decades later. In the 1950s, a Soviet biochemist named Boris Belousov was looking at the reaction of glycolysis, of how glucose gets broken down in our cells, in our bodies, in the process of metabolism. And he devised a mixture of chemical reagents that would mimic this process in a simpler way, in a test tube. So his reaction was a mixture that changed between yellow to begin with, and it went colourless as the reaction proceeded. But it didn't just stop there. It went back to being yellow again, and then it went to being colourless again, and then to yellow, and back and forth, and it just kept oscillating back and forth. It was an oscillating chemical reaction. Now, this seemed at the time to violate the second law of thermodynamics, and so other scientists weren't at all happy with this, and no one believed Belousov's oscillating reaction. He couldn't get the results published anywhere. He had to sneak them out in an obscure conference proceedings. It wasn't until this reaction was revisited in the 1960s by another Russian biochemist, Anatoly Zhabotinsky, that anyone started to take them seriously. And part of the reason for that was that Zhabotinsky found a way to modify this mixture to make the colour change much more dramatic. So in Zhabotinsky's version, it changed from being blue to red to blue to red. It just kept switching back and forth as the clock ticked on. And this was you could see this before your eyes. It was very hard to ignore the fact that this was a real process. And we now know that this so-called Belousov-Zhabotinsky reaction is just one example of a wide range of chemical processes that undergo oscillations. And in fact, the second law of thermodynamics isn't violated by them at all, because the oscillations don't go on forever. Eventually, they'll die away if you just leave this long enough, and it settles down to a steady state. And so that's all that thermodynamics cares about, what happens in the end. It doesn't say anything about how it gets there. These oscillations can be explained as a competition between two different intermediate states. So you can say a red one and a blue one that just switch back and forth. And the key here that enables this back and forth switching is that both of these processes have feedback that speeds them up. They're autocatalytic. The more, if you like, the more red stuff there is, the more it speeds up its own creation. And eventually it does so in a runaway process that just exhausts itself. There's nothing else to react. And so at that point, it's exhausted, and it's also set up the conditions for a switch over to the blue state. And then that runs through the cycle as well until it exhausts itself and switches back. So this feedback, this autocatalysis is essential to the process. Now, what's really going on here then is a competition between how quickly you can replenish all the ingredients you're using up by having them diffuse from further afield, and how quickly they react. It's a competition between diffusion and reaction. And this sort of system is now an example of what's known to chemists as a reaction-diffusion system. Now, if the reaction is well mixed, as I've shown here, then this colour change happens everywhere at once. But if it's carried out in a solution that's just left to sit quietly, particularly if you slow down the diffusion by actually putting it in a gel, like Liesegang did with his bands, then what happens is that the colour change can be initiated at different spots at random and then spreads from those in a kind of spreading wave. And because of these oscillations, those waves oscillate. They become a series of pulses, and what you get are these fantastic spatial patterns, these spiral and target waves spreading out like this. And these snapshots might put us in mind of the stripes of a tiger or a zebra, but although they're more regular, obviously. But the crucial difference is that they're moving patterns, whereas the animal markings are stationary. But it turns out that a reaction diffusion system can also produce stationary patterns. And it's very apt that this gathering is happening not only this year, but actually this week, because this happens to be the anniversary, 100th anniversary, the centenary of the birth of the man who figured out how you can get stationary patterns in a system like that. And this is Alan Turing. And in fact, in this week's Nature, there's an exploration of the legacy that Turing left us with. Turing's most famous for his work in decrypting the Enigma code that was used by the German military forces during World War II. He's also credited as being a founding father of artificial intelligence, sort of laying some of the foundational work of the whole field of computer science. But Turing's curiosity was very diverse, and in 1952, 2 years before his death, he published a paper in which he attempted to explain how symmetry gets broken during the formation of a body plan in a fertilized egg. And what I mean by symmetry breaking here is that you start off with a perfectly symmetrical, a uniform system, if you like, a ball of identical cells, and somehow some of these cells will decide, if you like, to become limbs and eyes and maybe a brain and so forth, and some will follow other developmental routes. And so the problem is how this initially uniform state spontaneously starts to develop these patterns that differentiate one part from another. And you can see that this is precisely the same kind of problem, or at least it seemed to be, that, that we find with the zebra stripes, how you, an initially uniform patch of skin, how it gets broken up into patches that get pigmented and patches that don't. Now, Turing's theory of how this could happen was quite abstract and complicated, and it was only in the 1970s that other scientists figured out what the crucial ingredients were. Turing suggested that the patterns could be created by two types of chemical substance, both of them diffusing through the system, and he called them morphogens, literally meaning shape formers. One of them was called an activator, and this was a stuff like as in the Broussard-Jabotinsky reaction. This was a substance that was autocatalytic, that created more of itself, that amplified itself. And the other one was an inhibitor, a substance that interfered with that process of self-amplification by the activator. And crucially, if the inhibitor diffuses at a rate that's faster than the activator, then you can get these spatial patterns starting to arise. Essentially what's happening is that you can get little local blobs of activator that sort of that amplify themselves and that create an enhanced concentration there, but that the inhibitor stops that process from spreading too far, and so you can only get another blob a certain distance away. Now, Turing had to do all his calculations by hand, and so it's hard to see exactly what the kinds of patterns would be that this activator-inhibitor scheme would generate. But when computers became available to do the number crunching, then it became clear that there are two generic types of pattern that this Turing process forms, and they are spots and stripes. But no one was able to find a real chemical system in which this happened until 1990, when a group in France managed to generate these patterns here, these genuine Turing patterns in a chemical system. And the following year, another group in Texas found a way to make them spread throughout an entire system. So these are not artificial colours, these are real chemical ingredients here at the bottom. Spots and stripes being formed in these very regular patterns, even though the ingredients, the molecules here, are diffusing throughout the whole system. And yet these stationary patterns remain. So Turing's theory did seem to offer, and still does seem to offer, the most plausible mechanism for many animal marking patterns. And the idea is that there are biochemicals that act like in activators and inhibitors, like these morphogens, during the growth of the embryo, and they imprint the skin with patterns that either switch on or off pigment-producing genes. And so these pigment-inducing morphogens would diffuse through the skin of the developing embryos and lay down that pattern. And Turing-type models of this process have now been put forward to explain many of these different animal markings that are actually more complex than the ones in the simple ones that I showed earlier on. So these network structures like you see on giraffes, and the kind of rosette structures that you see on jaguars and leopards, and the structures on ladybirds. One of the clearest examples of this sort of process is found on the angelfish. The angelfish is interesting because the patterns there aren't just laid down during growth and then just are fixed and sort of just blow up like an expanding balloon as the organism grows. They actually keep changing as the fish gets bigger. And you find in particular you get these kind of little zip stripes that gradually unzip as the fish gets bigger, and you see precisely the same kind of thing in a computer simulation based on a Turing-type mechanism. However, no one has found, for a system like this, no one has found the clinching proof, which would be to actually find the molecules that act as these morphogens. However, they do seem to have been found, or at least good candidates seem to have been found, in another kind of regular pattern that is formed in skin, and that is the formation of hair follicles. Hair follicles in mice, although it seems reasonable to imagine that a similar process is happening in humans. So this almost sort of regular, quasi-crystalline sort of structure that you see in hair follicles seems to possibly be laid down by Turing-type morphogens. There are several other candidates, biological candidates for these Turing patterns as well, and in fact, Richard and Anna have between them reminded me that another one is the formation of feathers themselves, the barbs of feathers, these very regular, if you like, a sort of regular system of stripes, seem quite plausibly to be a Turing-type pattern that involves an activator that I don't know if Anna's here, but it was her favourite molecule, this SHH molecule, the sonic hedgehog gene. Which seems to, I'm sure you all noticed in her diagram of the genetic network, there was a little loop, a little arrow going around on the sonic hedgehog in the middle, going back on itself. It's an activator. It's self-amplifying. So it seems quite possible that that's acting as an activator in the formation of feather barbs. But what then of this similarity between the stripes of a zebra and the stripes in windblown sand? Well, it's been argued that this generic process of interaction between a kind of localized activation, a self-amplifying activation, and a longer-range suppression or inhibition could be at work in the formation of a very wide range of natural patterns, and that this is one of them. Because if you think about it, you imagine a flat surface of sand across which wind is blowing with sand grains being carried in it. If by chance a little bump appears on that surface, just because a little bit more sand happens to be deposited there at some point, it sticks out above the rest and it starts to collect more grains. So the more it sticks out, the more grains it collects, and it's self-amplifying and it starts to grow. At the same time, by collecting those grains, it depletes the wind of the grains of grains in that vicinity. And so another bump can't grow until it's a certain distance away from that one, and so you get another one growing, then another and another in this more or less regular pattern. Well, this is basically what's going on. The actual process is a little more complicated than that. You have to take into account that the sand grains don't actually just stop where they fall. They perform a series of little jumps, and it seems to be the size of those jumps that sets the size of the ripples. And there are several other patterning processes going on at the same time in the desert, because we see several other features, several other pattern features growing on top of these larger scale structures, sand dunes. And these don't just appear as a series of sort of linear structures like the sand ripples. They have a whole variety of different shapes. So you can have these linear ones, or in fact in this case they're more sort of wavy. These are actually called seif dunes, and you see with the ripples superimposed on top of them. You can also get these crescent-like shapes called Barkhan dunes, or many-armed shapes called star dunes. And it turns out that if you alter gravity and alter the wind speed, which doesn't sound like an easy thing to do, but you can do it on Mars, then you find quite different shapes coming, sand dune shapes appearing. And this is what we find on Mars. This is a real picture of a strange kind of dune formed on Mars that we don't find anywhere on Earth. And all of these shapes can be reproduced in computer models of this process that are basically models of this activation and inhibition. Well, I think the key point here that I want to stress is that the patterns that we're seeing here don't respect the normal boundaries that we draw between the living and the non-living worlds. The patterns are governed by rules that are mathematical, not genetic, or even explicitly chemical. And as far as living nature is concerned, this implies that the constraints and the possibilities extend beyond the purely functional. Now, I'm not aware, as I say, that anyone will argue with the idea that the zebra stripes are an adaptation, but also no one will argue that they are a particularly finely tuned adaptation. That's to say exactly how the stripes fork and merge, how their width varies on different parts of the body, and why they're only roughly regular. All of these seem to be features determined more by the mathematics of the general scheme by which they're made, and not by the demands of evolution. So long as these features, these variations, aren't significant impediments to fitness, they can survive. They can even be toyed with and elaborated by nature without any obviously evolutionary function. And I'm reminded in all of this of what Vladimir Nabokov said about butterfly patterns. Now, you may know that before he became a famous novelist, Nabokov was a professional lepidopterist. He was actually curator at Harvard University's Museum of Comparative Zoology, and he knew an awful lot about butterflies. And like Darcy Thompson, he wasn't fully satisfied with Darwinian explanations for how they get their patterns. Nabokov suspected that there might be other imperatives at work. After all, this mimicry seemed sometimes to go much further than it had any adaptive reason to. This is what Nabokov said about it. He said, when a butterfly has to look like a leaf, not only are all the details of the leaf beautifully rendered, but markings mimicking grub-bored holes are generously thrown in. Natural selection, in the Darwinian sense, could not explain the miraculous coincidence of imitative aspect and imitative behaviour, nor could one appeal to the theory of the struggle for life, when a protective device was carried to a point of mimetic subtlety, exuberance, and luxury, far in excess of a predator's power of appreciation. I discovered in nature the non-utilitarian delights that I sought in art. Both were a form of magic. Both were a game of intricate enchantment and deception. Now, this all sounds perhaps a little too mystical, or even or a little too whimsical for some tastes. Nabokov has sometimes been accused of verging on almost a sort of creationist point of view. But there's something in his perception that butterfly wing patterns are not just the result of random doodles that chance upon some evolutionarily successful design. That perhaps instead they're variations on a theme, determined by the constraints of a pattern-forming process in which we can see intimations, at least, of Turing's mechanism. And now, it's pure anthropocentric whimsy that makes me illustrate that point this way. These are all natural patterns found on butterfly wings. But I hope that it will make you appreciate that, that nature seems able to do a lot with a few general rules. And the question that I want to leave you with is whether perhaps sometimes she contrives to do more than she strictly needs to. Thank you.
We can handle a few questions here. Can't see anybody. But.
This may have been mentioned earlier, but Weishaupt and Ruslan-Wohlhart worked out a lot of details of pattern formation from a genetics point of view in the fruit fly.
Yeah.
So has anyone followed the zebra during development to look at changes in RNA localization and protein? I'm not sure which proteins are involved, but maybe you could try and follow it over development.
Yeah. For the zebra, I'm not sure if they have. I think a lot of work now is going on in species like the angelfish and other pattern fish. I may be wrong, but I haven't seen any work specifically for the zebra to know what's going on there. But for the fruit fly, it's an interesting case because the fruit fly, in the early stages, the embryo develops a series of bands. And there are some people who looked at these and thought, Turing stripes, it must be. And it turns out that it's not. That's a system that is both, in some ways, both more complex and more simple than what's going on with Turing. In particular, because the stripes aren't identical. They don't sort of have identical fates. And so I think it was that realization that actually led to a bit of skepticism about whether Turing patterns have any role. Because here was an example where it really looked like they did, and then it seemed that they didn't. But I think in some of these other examples that I mentioned, it seems that there is a good case to be made that at least something like diffusing morphogens and autocatalytic feedback or self-activation does lie at the root of some of the patterning processes.
I wonder if you could speculate a little bit about Nabokov's whimsical comment about imitation. I mean, do you think that what we're seeing there is just parallel processes, or is there some interaction actually between the butterfly and the environment that it's on?
Yeah, Nabokov certainly knew. Whether I could speculate, whether I could amplify what Nabokov said about this apparent fidelity in the mimicry that some butterfly species show. And I'm afraid I probably can't. I mean, Nabokov knew an awful lot more than I did about this. Of course, a lot of the mimicry that we see in butterflies does have a very clear evolutionary function. That some butterflies are mimicking either poisonous species, where they're not poisonous themselves, or that there are other clearly adaptive processes that are going on, that they're sort of mimicking each other. When they're mimicking something, when it's sort of camouflage like this, I mean, Richard, I'm sure, will have much more insight into this than I have. I don't know what to say about Nabokov's view there. The mimicry is sort of much better than it needs to be. I don't even know whether he was right to say that. But certainly, from this outsider's point of view, you can see what he's driving at. That some of the mimicry of dead leaves and so on seems extraordinary. And it seems extraordinary, to a degree that you can't imagine a predator, needing in order to be confused that way. But I'm afraid I can't really say much more than that. I know there are other people here who I'm sure could.
One more from the front.
I have one question in regard to all the other papers we had in the morning session, and this is, first, I like your paper very much, and I think it brought another aspect in it, and I'm very curious to hear a little bit more about if you relate the stripes to Turing, and if I understand Turing correct, right? He's developing an idea which is completely independent from its environment, and the machine is constantly repeating and with that activating itself. That's very interesting because in the morning session we had heard a lot of papers that whatever the scientists were talking about, they related it to the environment and how relevant the environment is, also mimicry and so on, and everything we have heard for their theory. And we've once heard, for instance, the relevance of the Umwelt from Uexküll. Now, if I understand you right, do you vote for what we have with the stripes of the zebra is an autopoietic system which just develops it by itself without any reference to the environment?
No, not quite. I have no doubt that whatever a Turing-type mechanism or any other patterning mechanism might produce is then subject to selection, without a doubt. It's a question of how much selection. And I think that there are certainly some systems, whether this is true of the zebra or not, I don't know, I suspect not, but there are some systems like the seashells where the selection that it will be subject to is very weak, or perhaps non-existent. And in that case, it really is the case that nature, if you like, can play that all of these structures can come about, you know, without purely through their own dynamics. There's nothing that's going to be sort of, you know, winnowing them down. So I think that's really the question. There's always going to be, you know, for any organism, there's always going to be selective pressure, and the question is really how much. And if there isn't much, then, you know, you can see some interesting things going on that won't necessarily be Darwinian.
All right, sorry, we must move on. Thank you, Mark.